Hi guys, this video is going to be about translation. Before I start talking about the process of translation, I want to go over a couple of vocab terms for context. You know what mRNA is. That's messenger RNA, and we've been talking about how it's transcribed from DNA. tRNA is transport RNA. That's what the T stands for, transport. rRNA is ribosomal RNA. So that's what the R stands for, and that's what ribosomes are made out of, is rRNA. This is an example of tRNA. So each tRNA has what's called an anticodon on it, and there's an amino acid that is attached to it. So certain anticodons go with certain amino acids. This whole structure is actually made out of RNA. That's why it's called transport RNA. It's not made out of a protein or DNA or any other structure. This is all a bunch of nucleotides attached together in that structure. Anticodons recognize codons on mRNA strands. So remember, a codon is a set of 1, 2, 3 nucleotides. And when a tRNA recognizes that codon, that amino acid is added to the growing chain of proteins. So I'll show you a little bit more about what that looks like in a second. So a little bit more detail about rRNA, ribosomal RNA. So it's made of a large subunit and a small subunit. Sorry, things kind of got smudged here. Unit. So it's kind of self-explanatory. The large subunit is the big one, the small subunit is the little one. In each ribosome, in each set of ribosomal RNA, there are spots for tRNA in the large subunit, and there's a place for mRNA in the small subunit. So the purple structure is a ribosome. We've kind of just talked about them as organelles in a hand-wavy way, and then the mRNA goes to a ribosome and protein is made. Ribosomes are made out of ribosomal RNA. Again, it's all nucleotides in the structure that allow for this process to happen. So AKA, this is a ribosome. Just like with transcription, there are 3 stages of translation: initiation, elongation, termination. Same 3 stages, same 3 main ideas, just different things are happening because it's a different process. So in initiation, that also has 3 steps within it. The first is the small subunit binds to mRNA. So I tried to represent that here. So the small subunit of the ribosome binds to an mRNA strand. So that's our processed mRNA that's come out of the nucleus and is now in the cytoplasm somewhere interacting with this ribosome. The next step within initiation is tRNA with an amino acid attached to it binds to the start codon on the mRNA. So you guys have seen that AUG is the start codon and MET is the amino acid associated with that. So AUG is here on the mRNA. The anticodon is on the tRNA, so the opposite of that, the anticodon. So that would be UAC. So it binds to that, the tRNA binds to that small subunit that has the mRNA in it already. The last step in initiation is that the large subunit comes and attaches. So that large subunit of the ribosome joins the tRNA and the mRNA that have already interacted with the small subunit. So that was initiation. That's how it gets started. From there, we have elongation. So for elongation, come on, I'll just hold it. So for elongation, what happens is the small subunit of the ribosomal RNA, so that the ribosome pushes its mRNA a little bit further to the side so that it can see what the next codon is. So in this case, our next codon is GCC. So floating around in the cytoplasm are lots of tRNAs with their amino acids attached. When the right one, in this case CGG, so it has the anticodon of this codon, when it comes along, it binds in this spot in the ribosome. And when it binds, what the ribosome does is it forms a polypeptide bond between the protein, the amino acid from that first tRNA, to this amino acid that's just joined it, that's right next to it. So polypeptide bond is between amino acids. It's polypeptide bonds that create a protein. So these are the first two amino acids of our protein that's going to be created. All right, so that process continues. The tRNA that's already been used goes away, it breaks off, it's no longer attached to an amino acid, it's leaving the ribosome, it's going to go pick up another amino acid that matches with it so it can be used again. The next tRNA with its associated amino acid is going to come into this spot in the ribosome. Everything shifts down one. So in this case, we've already read AUG. We've already read AUG, that was this amino acid. Then we read GCC, that's this amino acid that's been attached to that first one. And now the next codon is UGA, so its corresponding anticodon is ACU. So whatever amino acid goes with this codon, you can look at your codon chart, would be attached to this tRNA. So that comes into the spot. The ribosome creates a polypeptide bond between that amino acid that's just been brought in and the one that was there before. And that continues over and over again as you grow a polypeptide chain, so aka growing a protein. So as these tRNAs come in, their anticodon matches with a codon. The amino acid that's attached gets put onto the one before it with a polypeptide bond. They get squished together, and the ribosome keeps reading. You get a protein. You get a polypeptide out of it. So that was elongation, that was extending the chain of the protein, of the polypeptide. So eventually we need to get to termination. So eventually the protein's going to be done. So the way that this works is when the ribosome reads a stop codon. So one example of a stop codon is UAA. The ribosome knows that it's time to stop translating. So this is a stop codon. So once that has happened, something called a release factor, which is a protein, comes into the ribosome. And what the release factor does is it tells the ribosome to let go of that polypeptide chain that it's been creating. So the release factor binds to the ribosome, and it tells the polypeptide chain that it can go. It tells the ribosome to release the polypeptide chain. So that's termination because that ends the whole process. So this was pretty good timing because I'm at the end of what I was going to say. That's the process of translation. As always, email me, text me if you have questions.